Hello everybody, and thanks for joining me today. So to briefly introduce myself, I've been leading the Research Service Division at Horizon for many years now, after co-founding the CRO division in 2007. I've worked in a number of biotech companies on multiple drug discovery programs and collaborations, and my core area of expertise is in oncology cell biology and providing expert contract research services to customers. So in the webinar today, I'd like to introduce you to Horizon Discovery and describe our functional assays division, focusing on how we use cell-based assays to aid our customers and collaborators to perform first-class oncology drug discovery. After a brief introduction to the company and to the Discovery Research Division, I'll cover applying cell-based assays to address key questions in cancer drug discovery, including the use of isogenic cell lines, compound and genetic studies, tumor microenvironment, and combination assays. And then I'll finish with some new developments in assay systems such as reporter assays and complex assays, including PDX and organoid cultures. So Horizon is a company heavily focused on using genomic information and translating this through gene editing and therapeutic understanding to personalized medicines. We're a products and services company and are able to support the whole continuum of the discovery pipeline, from basic research through to validated therapeutic targets. Cell line models can be accessed by multiple routes. We're well known for our expertise in generating specific mutation in customer cell lines through our custom engineering services offering. And we've also got a catalogue of over 500 isogenic lines, or else we can provide the reagents and design advice through our Genesis service also. And this enables customers to engineer their own lines. Target discovery and drug development services is the focus of this webinar. And this now includes our high-throughput combinatorics platform and in vivo models at our Sage Labs facility. Our diagnostics division provides genetically defined human genomic reference standards, including FFPE cell sections and purified genomic DNA. So Horizon services can be accessed individually or as an integrated project to enable us to support projects of any size. So Horizon is founded on the use of genome editing, and this is fundamental to our philosophy supporting oncology drug discovery. This slide is an old one, but it describes really nicely why we believe in gene engineering. Historically, to understand the effect of a specific mutation, classic overexpression studies have been performed, in which a mutant gene of interest is introduced randomly into the cell under the control of an exogenous promoter. This can result in unclear results due to the non-physiological levels of expression and regulation. For example, on the slides, we can see two studies investigating the effect of the same mutation. The first is the overexpression study, where PI3 kinase mutations are overexpressed, and this results in an oncogenic phenotype as shown in the left-hand panel. On the right-hand panel, however, these same mutations are introduced into the endogenous PI3 kinase gene, where physiological levels of expression and endogenous mechanisms of regulation are retained. And here, a much milder growth phenotype is observed, which alone would not be considered as transforming. Similar limitations can be seen with SI and shRNA studies, which attempt to knock down the expression of genes of interest. However, they're rarely complete knockdowns, and residual expression cannot be discounted, whereas permanent, stable disruption of genes removes such confounding issues. So generation of isogenic cell lines has been used to translate in vitro discoveries into patient treatments, and this slide shows how we have done this in a real example. So we use our Genesis engineering platform, where we can use IAV, zinc fingers, or the more newly described CRISPR-Cas9 nuclease to introduce any mutation of interest. It can be a point mutation, a knockout, deletion insertion, or whatever you're interested in really. An example here shows what we've done using the EGFR targeted agent Erbitux, otherwise known as cetuximab. So along the bottom, on the left-hand side, the in vitro proliferation assay data is using our isogenic cell lines and suggests that not all KRAS mutations behave in the same way to this drug. But currently, all patients with KRAS mutations are excluded from Erbitux therapy. But this in vitro data suggests that while G12V mutations are resistant, G13D might be responsive. This finding was then progressed into in vivo xenograft studies, where this result was confirmed. And retrospective analysis of clinical trial data suggests this could be a real effect seen in patients too, where patients with G13D mutations may benefit from Erbitux. And now new clinical trials are ongoing based on this data. Okay, so on to the Discovery Research Division. Horizon's services and collaboration platforms relies on 3 key tools to aid in cell-based assay research. The X-Man isogenic cell line system is a valuable tool in patient stratification, target validation, and for pathway interrogation. We use the tumor microenvironment via whichever assay is appropriate for the target and the biology we're interrogating. This could be 3D assays, it could be co-culture, hypoxia, or nutrient starvation. And we have 3 key genetic platforms that complement our cell line generation capabilities by using Si, Sh, or sgRNA. And in addition to this, we have all the typical offerings expected to support oncology drug discovery programs. For example, pathway analysis via Western's or ELISA, and live cell imaging using the IncuCyte Zoom. And this year, we've expanded our capabilities significantly with the addition of the high-throughput cellular combination assay platform at Combinatorics and the in vivo gene editing platform at Sage Labs. So at Sage, knockout rats and mice feature specific gene deletions, insertions, repressions, and also modifications. And these span multiple disease areas, including neurobiology, cardiobiology, oncology, as well as toxicology. But we'll cover this in a future webinar. Okay, so on to the body of the talk. So what are the key questions we address in our cell-based assay portfolio and each time we approach a project? One of the most important questions is the target well validated? We approach this question in a number of ways, and here I've highlighted just a few. So in the top left, we can use, here in this example, we have used shRNA to knock down the target in cells. This is an inducible, stable A549 cell line where doxycycline is used to induce knockdown of RAF1, resulting in an anti-proliferative phenotype. And here, this is seen with 2 independent hairpins. shRNAs are well known for having off-target and incomplete effects, so we've confirmed this result using the CRISPR-Cas9 system to deliver guide RNA to RAF1 and knock out the gene. The resulting anti-proliferative effect, in this case, is confirmed as shown in the top right. The benefits of the sgRNA approach are that target genes are disrupted and knocked out rather than causing partial depletion. In the bottom panel, this shows functional data from engineered cell lines from one of our target validation programs. Here we've knocked out our kinase target in HCT116 cells to generate an isogenic pair. The parental line grows well in the in vivo xenograft model, as shown in the blue line, but the identical cell line with the target kinase knocked out is no longer able to grow in vivo. This provides excellent validation for targeting this kinase. However, it does knock the target out, which is a different effect to that of a compound. Therefore, to definitively address the kinase activity of this target versus the scaffolding role, we engineered a point mutation into the kinase domain of the target. The red line shows the wild-type cell growing normally in 3D, with the green line showing 2 clones of the kinase dead cell line unable to grow at all in this 3D environment. So using one or a combination of these tools, we are able to provide robust data that validates a target before significant efforts are put into further project work. More recently, we've devised a medium throughput method that can shed light on ambiguous results that can emerge from SI, SH, or sgRNA screens. Ambiguous results can result from shRNA studies, as shRNA only gives partial knockdown, and greater phenotypes are often partial too. In sgRNA screens, repair of Cas9-mediated double-strand breaks can result in in-frame indels that don't disrupt protein function. So in this new system, we would use lentivirus to deliver Cas9 and sgRNA to the target cells, allow time for gene editing to occur, and then culture colonies from single cells. Horizon's cell line engineering expertise allows us to devise an analysis pipeline to characterize editing events on the clonal level. If viable colonies contain frame shift indels, this tells us the target is non-essential. But if viable cells contain only in-frame indels, the target is confirmed as essential. So on to the next key question we often face, which is, which patients should I target? We can investigate using our isogenic cell line panels. Whereas non-isogenic cell lines contain multiple different genetic differences, isogenic cell lines vary only in the target of interest. As I previously mentioned, we have a large catalogue of lines, as well as an expert scientific team able to engineer cell lines to our customer specification. So this study is designed for a customer who had a selection of compounds against various targets, and they were looking for specific indications to follow for a route to the clinic. We tested around 200 compounds in a panel of 30 isogenic cell lines. Each panel shows the same compound in different lines of the same background parental cell line, and you can see the various effects, with some compounds being inactive and some active in all lines tested. The interesting results were clear to see. In the top left panel, this compound is equipotent in most of the lines except one where it shows a clear sensitivity. Similarly, in the bottom panel, this compound is inactive in most of the lines except for one, where it kills the cell line harboring a key oncogenic mutation. This data from panels of cell lines containing patient-relevant genotypes gives valuable insight into biological mechanisms and interactions to follow for a path to the clinic. Another way in which we can investigate which patients to target is to perform synthetic lethality screens. In particular, performing such screens using isogenic cell lines allows for a much more precise system and removes a key source of noise when using cell lines with multiple genetic differences. So here, we have used our siRNA library of around 2,000 genes in an HCT116 isogenic pair. Each individual siRNA is plotted along the x-axis and the sensitivity along the y-axis. The blue line represents the effect on the parental cell line, and the green dots the effect of the mutant-matched line. As the mutant response pulls away from the parental response, we see a set of genes of high interest as highlighted. The larger the dot, the greater the significance of the differential response between parent and mutant line. And then we can follow these hits in validation studies to identify new patient populations to study. So the converse question is an equally important one: which patients should I avoid? So similarly, using isogenic panels, we can define patient populations that might be resistant to a given drug. The example shown here is of a tyrosine kinase inhibitor. The SW48 colon cancer cell line is sensitive to the drug, which is shown as the parental line in red. But when it's tested in matched cell lines with mutations in PI3 kinase or P10 deleted, clear resistance is seen. Right, okay, so back to the sort of questions we're asking. And the next one I was going to cover is my assay configured correctly? So this example shows the well-known effect of the PARP inhibitor oliparib on parental DLD1 cells versus the same cell line where the BRCA2 gene has been silenced. In a standard 96-hour proliferation assay, the increased sensitivity can be seen in the BRCA null line compared to the parental. But this is a very well-developed compound, and seeing these kinds of differential responses in earlier stage and less potent compounds can be problematic. So in this case, a much more appropriate assay would be a long-term colony formation assay, and we run these medium throughput assays routinely. Here we can see the effect of oliparib is now not only significantly more penetrant, but also the difference is significantly greater in magnitude. An example shown on this slide is looking at the bioreductive drug tirapazamine. This drug requires hypoxic conditions as present in solid tumours to become activated. Using standard conditions compared to hypoxia of 1% oxygen shows a small activation of the drug. That's shown in the left-hand graph from the grey line to the dark orange. But a much more prominent effect is seen when you're using anoxic conditions, which is then the leftward shift in pale orange. So by being able to dial in different oxygen concentrations, we can validate our assays and determine the appropriate conditions to study specific agents. We've also used a 3D spheroid assay system to study tirapazamine. The top photograph here is of a tightly packed sphere of HCT116 cells, and the bottom image is a cross-section of that spheroid with the hypoxic core highlighted. Multicellular tumour spheroids offer many advantages over traditional 2D cell culture systems, including the ability to gain insights into therapeutic problems associated with metabolic and oxygen gradients, which are commonly found in tumours, also to address cell-cell and cell matrix interactions, and these may have influence on chemo-resistant mechanisms. So using this spheroid system, we're able to see clear activity of the bioreductive drug, as shown in the middle graph. This is a much more relevant assay situation than 2D, where the hypoxic core has been defined both by the staining and by the drug response. Okay, this leads nicely onto another area of focus, which is to consider the impact of the tumour microenvironment. So different targets and pathways have different requirements on the tumour microenvironment, and studying under more appropriate in vivo-like conditions can be key to interrogating your target appropriately. The example shown here focuses on KRAS. So using our model DLD1 system, we have shown a requirement for 3D culture to reveal the underlying dependency of the cell line on the KRAS mutation. DLD1 cells contain an endogenous G13D mutation. So using our engineering technology, we have knocked out the mutant allele of KRAS. So in this system, both cell lines grow identically in 2D, as shown in the top left graph. However, once cells are grown in 3D soft agar system, the KRAS mutant line is unable to grow well, while the line with the KRAS mutation knocked out is no longer able to sustain growth. This is demonstrated in the right-hand graph. So this is further validated using both siRNA and compounds. And this is shown at the bottom. So we've developed an siRNA soft agar protocol and are able to screen medium throughput siRNA libraries in soft agar. Here the image shows siRNA to KRAS, and it ablates the growth phenotype of the cell line in soft agar. Similarly, when looking at pathway inhibitors, these two MEK inhibitors show relatively poor anti-proliferative effect in 2D culture, but when using the same cell line in 3D, we're able to see a marked dependency on the pathway and clearly determine the drug effects. Okay, so the final question we routinely ask is, what about combination therapy? So Horizon recently acquired the Combinatorics high-throughput combination screening platform. Here we're able to screen over 800 cancer cell lines, and our isogenic panels are being incorporated into this as a further set of lines. Routinely, 250 enhancer agents are screened in these panels with customer compounds. The team at Horizon Combinatorics has over a decade of experience in drug combination screens and its proprietary Chalice software for the analysis. Combination therapies are becoming standard of care in indications such as cancer, and identifying novel multi-target mechanisms that better address the system biology of disease at an early stage is invaluable information for any drug discovery program. Further information on the Combinatorics platform will be presented at a later webinar, but if you have a specific interest, then please let us know and we can follow up directly. So I've described these cell-based assays as standalone systems, but of course they form part of an integrated solution. This flow diagram shows just one way in which our customers and collaborators can work with us. So this example shows a client wishing to start with a large-scale profiling project and then to narrow down the mechanism of action and validation. So following a large-scale cell screen and combination study, hits are nominated and progressed into further studies. These can be secondary assays, such as higher order combinations where more than 2 drugs are combined, using microenvironment conditions such as 3D or co-culture. They can be interrogation of mechanism of action by genetic characterization or biomarker work, and can include gene expression analysis. Furthermore, isogenic cell lines can be made for definitive target confirmation, and different in vivo models are offered through engineering standard xenografts or PDX models. Customers can access our services at any point, and of course a project can follow any direction as well, and we work closely to devise the right study to answer the question. So I'd just like to finish on some recent developments and new assay offerings we're working on. An exciting new area that is gaining a lot of interest at the moment are endogenous reporter lines. The same limitations that apply to overexpression studies apply to systems employing synthetic reporter systems. So in collaboration with Promega, we have generated panels of cell lines that contain genes endogenously tagged with either the multi-purpose halo tag or nanoluck, an extremely bright reporter. The left-hand panel shows the halo tag system. Here, SW48s have been engineered so that endogenous KRAS has been tagged with the halo tag. After adding the ligands, KRAS localization can be followed in real time. The right-hand panel shows one of our suite of nanoluck lines. Here, HCT116 cells are the background, and MYC has been targeted with nanoluck, and signal dynamics followed after treatment with the transcription inhibitor actinomycin D. An excellent signal-to-noise dynamic is shown for the luciferase readout in red, and the blue line is the viability readout via cell titer blue, and this shows no impact on cell viability. So using this technology, it's possible to monitor the regulation of gene expression at the endogenous level, and these nanoluck lines are being used in high-throughput screening formats. We're also investigating more complex disease models in the form of both PDX and organoids. Through Sage Labs, we are establishing 25 breast cancer PDX models, which were originally developed in the labs of the University of Washington. These are one of the most characterized sources of breast PDX models in the world, and include 10 triple-negative lines. We're also very interested to explore the possibility of growing iPS organoids, both for screening and for cell line engineering purposes. The data shown here is from our lab, where we are now at the exciting stage of sectioning fully formed organoids. So finally, we've been investing heavily in the establishment of new sgRNA screening service. You can find out more about this in our next webinar, which is focused on synthetic lethal target ID using RNA-based screenings, which is scheduled for next month. Okay, so my final slide returns to the drug discovery pipeline and illustrates the areas in which Horizon Discovery Services can help you, whether it's at the early target ID and validation stage using our screening and cell line generation capabilities, in hits to lead or lead up with mechanism of action support, in vivo models or combination screening, or at late stage patient stratification or drug repositioning to profile compounds in isogenic lines or interrogate new indications through combination screening.